Hello, everybody. My name is Mr. Amador Hernandez, and today I'm going to be presenting to you the congenital anomaly of the sign, which is maternal PKU, or in other words, maternal phenylketonuria. And the paper I chose for my assignment was Seagreaves and McBride's paper entitled "Cardiac Teratogenicity in Mouse Maternal Phenylketonuria: Defining Phenotype Parameters and Genetic Background Influences." So, before we begin talking about MPKU, let's address the PKU syndrome. So PKU is defined as a deficiency in the activity of the phenylalanine hydroxylase gene. So this hydroxylase gene is responsible for phenylalanine hydroxylase enzyme, which is responsible for converting phenylalanine into tyrosine. Tyrosine is an important amino acid in the body because it's important in protein synthesis for proteins like melanin, catecholamines like epi, norepi, and dopamine in the brain. In the case for PKU individuals, unfortunately, they cannot make this enzyme, and so phenylalanine is never converted into tyrosine. Instead, it's built up along with metabolites in the body and cause associated health problems. The outcomes of PKU is consistent with intellectual defects, immature growth of vital organs such as the brain and the heart, and an increase in mortality of the newborn. Three types of three types of PKU do exist, and they differ based on their levels of phenylalanine concentrations. So, in the first case, hyperphenylalaninemia usually results in a blood concentration less than or equal to 600 micromoles per liter. In classic PKU, we see that blood phenylalanine levels are greater than 1200 micromoles per liter, and in variants such as atypical PKU, they range from 600 to 1200 micromoles per liter. Well, one thing to note is that normal phenylalanine levels are usually less than 360 micromoles per liter, which is usually recommended to maintain healthy pregnancy and ensure minimal health complications to the neonate. So, in terms of severity, it is fair to assume that hyperalaninemia is the most mild case, and classic PKU covers more severe cases. So, now that we have an understanding of the underlying complications associated with PKU, we now take a closer look and define maternal PKU, which refers to the teratogenic effects of PKU during pregnancy. For those not familiar with these effects, they're just simply any effects that cause an increased chance of abnormality among fetal development. And so best observed outcomes occur when strict control of maternal blood phenylalanine concentration is implemented before pregnancy or by 8 weeks of gestation at the latest. PKU occurs in 1 out of 10,000 to 50,000 live births and is most often diagnosed using the newborn screening program in Ontario, which involves taking a sample of blood from the baby's heel after birth, and the sample is then taken to the Ontario Newborn Screening Program in Ottawa for analysis. So MPKU does follow an autosomal recessive pattern, which requires two abnormal alleles at the PAH or the phenylalanine hydroxylase locus. And so I designed the pedigree just for visual representation, and it shows that the mother with PKU, dark blue, has two abnormal alleles, and then the father with no PKU has one abnormal allele, making him a carrier. And so in this case, there's going to be a 50% chance that each offspring will either have PKU or not have PKU and be carriers. And this is because despite the genotype of the father, the mother will give every single offspring at least one abnormal allele, thereby making it a 50% chance of them either receiving an abnormal allele from their father as well, giving them PKU, or receiving the normal allele from the father, making them carriers. So now we're going to talk about what is the standard treatments for mothers with PKU. In that case, it's the phenylalanine restricted food diet. This diet contains foods that are lower in protein because foods that are usually high in protein are often high in phenylalanine as well. So the diet usually consists of lots of fruits, veggies, low protein breads, etc. But to support normal growth development, tyrosine still needs to be supplemented to perform the synthesis of proteins like melanin and catecholamines. And so these medical foods come in the form of amino acid mixtures, similar to like a protein powder mixture, and that contain tyrosine, of course, and then specialty drinks and snacks. So, clinicians do recommend that the special diet is maintained for life, as it's the best course for preserving intelligence and cognition. Enrolling mothers into counseling and prenatal care programs is important for optimal fetal outcome. And this is because it provides recommendations for what measures the mother needs to take based on her phenylalanine blood levels, and also educates the soon-to-be mother on the overall risks associated with MPKU. So here's just an image of expecting parents taking a prenatal education class. And not as for potential therapies, but to note is that they're still being determined to see if their role in reducing the risk of MPKU syndrome is significant. And I'm just going to be talking about one for today, and that's the large neutral amino acid supplementation, which involves using large neutral amino acids to outcompete with phenylalanine for transport across the blood-brain barrier, thereby decreasing the concentration of phenylalanine and its metabolites in the central nervous system. Other therapies include BH4 administration and enzyme therapy. So before we begin discussing CVMs or cardiovascular malformations in mouse, let's talk about in humans for a second. So one thing I do want to note is that you're going to be hearing a lot of terms you may not be familiar associated with the heart, but no need to worry. I just mentioned them to kind of provide a trend. Not so much attention is paid to the actual defect. Just focus on the take-home message I'm trying to portray. So MPKU is associated CVMs occur in 7 to 50% of cases, with the most common defects being the coarctation of the aorta. So it just simply refers to the narrowing of the aorta, and therefore this causes obstruction to blood flow and leading to cardiac complications such as ventricular hypertrophy, so that's the enlargement of the ventricular walls. And in the second defect, we have HLHS, which is a birth defect in which over here, you see that in the left, the whole left side of the heart is underdeveloped, including the left ventricle, the aorta, the valves, etc. And this causes numerous heart conditions. So within the last decade, the number of babies born to mothers with PKU is now estimated to equal the number of babies born affected with PKU. And this shows that MPKU is now becoming a significant health problem. So in the paper, authors aim to grasp a better understanding about the pathogenic effects of phenylalanine as a teratogen in mice so that further research can be performed to develop novel therapeutics in humans with PKU. So in this experimental paper, research aimed to address the following hypotheses. The first being CVMs occur in a dose-dependent manner, genetic background affects CVM phenotype, and exposure of high phenylalanine during specific times in heart development causes specific types of CVMs. So in addressing the first hypothesis, the mice model used in this experiment is the BTBR PAH NU2 model. So BTBR referring to the background of the mice, PAH refers to the mutation it has, and NU2 refers to the model. And so with this mutation, there's a missense mutation in the active site of the enzyme, rendering this mice with PKU. And to answer the hypothesis, the homozygous in females were bred with homozygous wild-type males to produce heterozygous BTBR females to avoid any effects of PKU in fetal animals. So 207 of these mice were screened for CVMs. But to do this, 4 treatment groups were established to investigate the critical maternal serum levels, which, so in the control, we have less than 360, low 360 to 600, in the mid, we have 600 to 900 micromoles per liter, and then in the high, we have more than 900 micromoles per liter. So serum phenylalanine levels were measured by an amino acid analyzer, whereas the blood phenylalanine levels were measured by draining the blood from the veins in the face of the mice onto a filter paper and then subjected to mass spectrometry for analysis. To note, CVMs were classified using the classification scheme, which were based on cardiac phenotypes, cardiac complexity, and extracardiac anomalies. All mice were placed on a phenylalanine deficient diet and given water containing phenylalanine at varying concentrations to maintain groups. The screening results are depicted in this table, which shows a positive correlation because as the groups varying concentration of phenylalanine increases, we also see an increase in percentage of fetuses with CVMs. So this picture, so this confirms that the CVMs occurred in a phenylalanine dose-dependent manner. In addition, many of the resulting CVMs are those affecting the outflow tract. So that is this over here, which are defects to the pathway in which the blood takes from the ventricles to the great arteries. So that's over here, so the aorta and other arteries from here. And so this is the normal morphology of the OFT, so the outflow tract. And then here we see abnormalities. So you don't really need to know what these are, but you see that the LCCA branches from the RSA and this abnormal image. But in reality, it's supposed to branch from the RIA. So, and then over here you see a similar trend as well. So abnormality in the origin of branching. So now when addressing the second hypothesis, congenic mice were created by breeding the PAH and U2 locus onto two different congenic mice. So it's this one over here and this one over here. To investigate the effects of genetic background upon incidence and phenotypic variation of cardiovascular malformations. So each strain was placed in similar conditions as seen in the previous slide. And in the case of C3H congenic mice, a dose-dependent manner was observed with higher incidence of CBMs than the other two strains. Common associated defects include subaortic valve stenosis and ventricular hypertrophy. In BL6 mice, congenic mice, they began to experience a dose-dependent manner, but begins to plateau after mid-levels of phenylalanine and require less phenylalanine intake to achieve higher serum levels. In other words, they're really susceptible to phenylalanine levels. And common defects include interrupted aortic arch type B. And from earlier slides, we know that the BTBR mice respond in a dose-dependent manner. And common defects include aortic branching, abnormalities as seen in the previous slide, and double alert right ventricles. But that's not really important what I'm trying to say. What is important, though, is that as patterns in dosing, incidence of CBMs, and associated defects differ between mice strains, it is fair to assume that differences in genetic background influence the type of CBM. So now we discuss the final hypothesis. That involves the question, does high exposure of phenylalanine induce CBM during specific stages of development? So to answer this, groups of 3 homozygous mutant females were exposed to a high phenylalanine diet for time periods staggering over embryonic days 7.5 to 15.5. Each group was given a high phenylalanine diet to elevate maternal serum levels at specific times during gestation. 1 day of no phenylalanine intake was given after phenylalanine exposures to rapidly decrease maternal phenylalanine levels to that of the control groups. And it was found that receiving high exposure between embryonic days 8.5 and 13.5 was enough to cause cardiac vascular malformations affecting the outflow tracts, which refer to the path in which the blood takes from the ventricles to the great arteries in BTBR mice. So previously I mentioned that the best observed outcomes for delivering a healthy baby with from a mother with PKU occurs when strict control of phenylalanine levels is taken by 8 weeks of gestation at the latest. So the data correlates well with human PKU, as embryonic day 14.5 in mouse is equal to 7.4 weeks, which is consistent with the point in human development where the embryo is extremely susceptible to CBMs as well, which demonstrates BTBR as a reliable model for PKU. Unfortunately, a cause for CBMs remains unclear, but there is strong support for cardiac neurocrestal abnormalities being the cause of CBMs due to phenylalanine teratogenicity. In both human and mice, migration of cardiac neurocrest cells from the pharyngeal arches, which are over here, I like to think of them as like the segments of Drosophila because they give rise to body parts. So they migrate into the arterial pole, which forms the base of the aorta and the pulmonary trunk, and this occurs from embryonic days 8.5 to 9.5. And this lies consistent with the data gathered because defects affecting the aorta is extremely common as we previously saw. Also, the timing in the migration of cardiac neurocrest cells is compatible with the sensitive period where fetuses is more susceptible to CBMs. In addition, during heart remodeling, which takes place during embryonic days 12.5 and 15.5, cardiac neurocrest cells are responsible for outflow tract septation. And this ensures that the blood moves in the proper direction throughout the heart, and it further helps in the development of the aorta, highlighting the importance of cardiac neurocrest cells in heart development. Although promising, additional studies are necessary to understand the role of cardiac neurocrest cells in the development of CBMs and PKU. So, to summarize the major findings of what were previously spoken about, we first learned that MPKU-associated CBMs occur in a dose-dependent manner. In addition, fetuses were sacrificed at embryonic day 18.5, giving researchers an idea of the phenylalanine level required to cause CBMs, which will be necessary for designing cellular assays of phenylalanine teratogenicity in future studies. Secondly, we addressed the influence of genetic background on CBM phenotype and found that all strains yielded inconsistent results, thereby proposing that differences in genetic background do influence the type of CBMs. Since all experiments prior used mice from the BTBR background, it was important to determine the phenotypic differences in MPKU amongst other enriched traits. And lastly, we saw that those fetuses that received high exposure between embryonic days 8.5 and 13.5 were exposed to enough phenylalanine to cause CBMs. Furthermore, results may be used to better understand the most likely mechanisms involved. For example, isolating the time of high exposure that leads to CBMs in the BTBR model will aid in determining the cell population, genes, and possible genetic pathways affected by phenylalanine. And so that concludes the end of my presentation. Thank you so much for listening. Stay safe, stay healthy. Have a great day.